Today I am taking up respiration in plants or cellular respiration part 2. In part 1 we have studied about glycolysis. Glycolysis, I have told you, occurs in the cytoplasm of all the cells in all the living beings, and the glycolysis is also known as EMP pathway after the discovery of glycolysis. E means Emden, M means Meyerhoff, P means Parnas. By taking the first letter of these three, the names of the three scientists, it is called EMP pathway. So we have known that, we have studied that the end product of EMP pathway is pyruvate. The fate of pyruvate depends on the availability of oxygen. If there is no oxygen, it undergoes anaerobic respiration. It is also known as fermentation. If oxygen is available, it enters aerobic respiration. First, I will take up anaerobic respiration. Anaerobic respiration is very inefficient type of respiration. Only 7% of the energy present in the glucose is harvested through this process, and the remaining energy is wasted or lost. This anaerobic respiration is also two types. In one type, ethanol is produced, and in the other type, lactic acid is produced. In the case of yeast, the pyruvate undergoes cleavage with the help of the enzymes pyruvic acid decarboxylase and dehydrogenase. So, these enzymes reduce pyruvic acid or pyruvate into ethanol and carbon dioxide. In this way, ethanol, it is alcohol, ethyl alcohol, and carbon dioxide is formed. Carbon dioxide escapes into the atmosphere. Ethanol concentration, if it increases to 13%, it kills the yeast. Then, the alcohol contains still lot of energy. So nowadays, this alcohol or ethanol is used in petrol or mixing with petrol and used as a fuel for driving motor vehicles. When ethanol is mixed with petrol, it is known as gasohol. In countries like Brazil, lot of ethanol is used in making gasohol and driving the cars. So, by making use of the ethanol as a motor fuel, the energy still locked up in ethanol can be utilized in a better way. India is also now trying to use energy present in ethanol for motor vehicle as motor vehicle fuel. The second one is production of lactic acid. In the case of bacteria and mammals, what happens is, if there is oxygen debt, that means when the metabolic rate is very high, enough oxygen is failed to be supplied to the tissues. So as a result, oxygen debt builds up. In that case, the pyruvate cannot enter the Krebs cycle because of the shortage of oxygen. Then the pyruvate is converted into lactic acid. In the previous class, by mistake, I told pyruvic acid is lactic acid. In muscles, lactic acid builds up. And the buildup of lactic acid causes muscle fatigue. Then, the enzyme involved here is lactate hydrogenase. Because of this enzyme, lactate hydrogenase, pyruvate combines with NADH and hydrogen ions and forms lactate in the liver. So this lactate is transferred to the liver, where it is reconverted back into pyruvate, or it is even converted into glucose. In this way, the pyruvate, the energy present in the pyruvate is not lost as in yeast, but it is reused in converting the lactic acid into a pyruvate or even into glucose. Then, if there is oxygen available, the pyruvate enters aerobic respiration. In all the animals, the rate of metabolism is very high. So to supply enough oxygen, in most of the higher animals we find respiratory systems to transport oxygen to the tissues directly. Coming to the plants, the metabolic rate is comparatively less than those of animals. So, that much oxygen is not required in the case of plants. Plants have so many other mechanisms to obtain oxygen for aerial respiration. You know that in photosynthesis, oxygen is released as a byproduct. That oxygen is used in aerobic respiration. Then there are lenticels and stomata through which plants obtain oxygen. Then coming to the living tissue in plants, the living tissue is mostly confined to the leaves, which are very thin and exposed to air, and also only to the surface areas of stems and roots. The inner part of the stems and roots is made of dead tissue, and the dead tissue mainly provides mechanical support. But the living tissue is only peripheral, and that peripheral tissue can obtain easily oxygen from outside. Then the parenchymatous tissue present in the living tissue is also loosely arranged, having so many gaps between the cells. So through these gaps, oxygen can be easily circulated to various parts of the plant body. So in this way, in both plants and animals, oxygen is obtained for aerobic respiration. In the case of aerobic respiration, what happens? The pyruvate combines with, and even before the aerobic respiration is carried out, pyruvate combines with coenzyme A and forms acetyl-CoA. During the formation of acetyl-CoA, one carbon dioxide molecule is removed and NAD is reduced to NADH and H. So in this way, two molecules of NAD is reduced. NAD means nicotinamide adenine dinucleotide. Then acetyl-CoA enters the Krebs cycle. It first combines with oxaloacetic acid. So acetyl-CoA combines with oxaloacetic acid and forms citric acid. That's why this cycle is also known as citric acid cycle. In this reaction, what happens? Coenzyme A is released from acetyl-CoA and it is recirculated like this. Acetyl-CoA is available in limited supply. So, the acetyl-CoA is recycled, coenzyme A. The coenzyme A is available in limited supply. So, the coenzyme A released in this reaction is recirculated again and again in this reaction or in the formation of acetyl-CoA. Then, during the conversion of acetyl-CoA into citric acid, one water molecule is consumed. Then, citric acid is the starting point of this one and it is converted into cis-acotinic acid. In this process, one water molecule is lost. Then cis-acotinic acid is converted into isocitric acid. Here again one molecule of water is consumed. Then isocitric acid is converted into oxalosuccinic acid and oxalosuccinic acid is converted into oxalosuccinic acid is converted into alpha-ketoglutarate acid. In this process, what happens is two hydrogen ions are removed and they are accepted by NAD or hydrogen accepts the nicotinamide adenine dinucleotide. So by losing two hydrogen ions, oxalosuccinic acid becomes alpha-ketoglutarate acid. Alpha-ketoglutarate acid is converted into succinic acid. In this stage, one water molecule is added, one carbon dioxide molecule is removed, two hydrogen ions are removed, and they are accepted by NAD, and the ATP is converted into ATP. One ATP molecule for each pyruvate is converted into one ATP molecule. In this way, total 2 ATP molecules are produced for one glucose molecule. Then, the succinic acid is converted into fumaric acid. In this process also, 2 hydrogen ions are removed. But remember carefully, these hydrogen ions are not accepted by NAD, but FAD, flavin, adenine, dinucleotide, and FAD is reduced to FADH plus hydrogen ion. Then, fumaric acid is converted into malic acid, and malic acid is converted into oxaloacetic acid again. In this process, again, 2 more hydrogen ions are released, and these are accepted by NAD. Because the reactions occur in a circular fashion, it is known as Krebs cycle. So the oxaloacetic acid is again reused in the process. So the important point about the cycle is oxaloacetic acid, which is present at the beginning of the cycle, is regenerated again at the end of the cycle for reuse. In this way, the Krebs cycle takes place in the mitochondrial matrix. I have told you, glycolysis or EMD pathway occurs in the cytoplasm of the cell. Then the pyruvate is transported into the mitochondrial matrix. Inside the mitochondrial matrix, all these reactions takes place. Now let us see how many NADH, FADH and ATP are produced in the glycolysis and also Krebs cycle. In the glycolysis, 4 ATP molecules are produced and 2 NAD molecules are reduced. During conversion of pyruvate into acetyl-CoA, 2 NAD molecules are reduced. Then, during the entire process of Krebs cycle, 6 NAD molecules are reduced. See, this is 1, this is 2, this is 3. 3 multiplied by 2, 6. FAD. 2 FAD molecules are formed. Then ATP. 2 ATP molecules are formed. This is 1 ATP. In this way, NAD, FAD and ATP molecules are synthesized during EMD pathway and citric acid cycle. These reduced NAD and FAD, what they do is, they immediately hand over the hydrogen ions to the hydrogen acceptor present in the electron transport chain. Again, there is a limited supply of NAD and FAD. So if they are locked up permanently, there will be no more FAD and NAD available for accepting the hydrogen ions released in the cycle. So the NAD and FAD, what they do is, they immediately hand over the electrons to the complexes present in the electron transport chain. That we will see now. In the mitochondria, this is the outer membrane, this is the inner membrane. Inner membrane is folded and produces crystal. The inner membrane contains electron transport system or electron transport chain, this one. The Krebs cycle is taking place in the matrix here. So for this membrane, this is the mitochondrial matrix and this is the intermembranal space. Between the outer membrane and inner membrane there is a space known as intermembranal space. So here the matrix, intermembranal space and the inner mitochondrial membrane are all important. Coming to the electron transport system or electron transport chain, there are 4 major complexes. 1, 2, 3, 4. The first one is known as NADH dehydrogenase. Then this NADH, reduced NADH, comes here and hands over the electrons to this complex 1. That means here what happens is it's converted into NAD plus 2 electrons, 2 hydrogen ions. NAD plus. Then these electrons are handed over to the complex 1. These hydrogen ions are pumped into the intermembranous space. From the mitochondrial matrix, the hydrogen ions enter the intermembranous space. Then the complex 1 hands over the electrons to the ubiquinone. Ubiquinone is attached or it is close to the outer surface of inner membrane. Outer surface of the inner, this is the outer surface of the inner membrane. This is the inner surface of the inner membrane. Remember, this is the outer surface. This is the inner surface of the inner membrane. So the complex 1 hands over the electrons to ubiquinone, which is close to the outer surface of the inner membrane. Then FAD. FAD hands over its electrons to the complex 2. Again the same thing happens. 2 electrons and 2 hydrogen ions are released. The hydrogen ions are pumped into the intermembranous space and the electrons are handed over to the ubiquinone. In this way, NAD and FAD hand over their electrons to the complex 1 and 2 respectively. In this process, the FAD and NAD are released and they are reused again in accepting hydrogen ions that are released during Krebs cycle. Then these electrons are handed over to complex 3. The complex 3 also expels hydrogen ions into the intermembranous space. Actually, these hydrogen ions are released into the intermembranous space through this complex. Then the electrons are handed over to cytochrome C. Cytochrome C is also close to the outer surface of the inner membrane. From cytochrome C, the electrons are handed over to the complex 4. The complex 4 also expels 2 hydrogen ions from the mitochondrial matrix into the intermembranous space. In this way, in this process, if it is NADH, 2 altogether 6 hydrogen ions or 3 pairs of hydrogen ions are pumped from mitochondrial matrix into the intermembranous space. In the case of FAD, only 2 pairs of hydrogen molecules hydrogen ions are pumped into the intermembranous space. That is the main difference between NAD and FAD. So, NAD causes pumping of 3 pairs of hydrogen ions into the intermembranous space, and whereas FAD causes only pumping of 2 pairs of hydrogen ions into the intermembranous space from mitochondrial matrix. That's why NAD produces 3 ATP molecules and FAD produces only 2 ATP molecules. I will explain that later on. The complex 4 is also known as cytochrome oxidase. Here, the electrons come out into the mitochondrial matrix. Then you can ask why the electrons are transported like this through the electron transport chain. If they're free, what happens? They combine with protons, hydrogen ions, and hydrogen atoms are formed, and that has to be prevented. So to prevent formation of hydrogen ions, these electrons are transmitted from complex 1 to complex 4. So in this way, now what happens at the end of this process? Hydrogen ions are in higher concentration in the intermembrane space and a lower concentration in the mitochondrial matrix. And this results in chemiosmosis. There is a concentration gradient. Hydrogen ion concentration is higher here and lower here. So the concentration gradient of hydrogen ions is established in the mitochondria. Now, the electrons coming out here combines with hydrogen ions and half O2 and forms water. In this way, cytochrome oxidase helps in the formation of water and also uses oxygen. So it is known as cytochrome oxidase, and in this way, metabolic water is formed. And this metabolic water is formed in the mitochondrial matrix. Then, these hydrogen ions released into intermembrane space, they do not remain there. They enter the mitochondrial matrix because of the hydrogen ion gradient. And these hydrogen ions pass through one enzyme known as ATP synthetase enzyme. This ATP synthetase enzyme is also known as complex 5. Because of the high concentration of hydrogen ions here and low concentration here, they pass through this complex 5 or ATP synthetase. We have studied this in the photosynthesis also, in the form of chemiosmosis. So, at a time, a pair of hydrogen ions pass through this ATP synthetase. How many? A pair, 2 hydrogen ions. As they pass through this ATP synthetase, which has again 2 parts, FO and F1, just like in photosynthesis. These are called F1 particles. And the ATP synthase is present in the F1 particle. And as the hydrogen ions pass through this one, ADP is converted into ATP. That means for each pair of hydrogen ions, one ATP molecule is synthesized. Remember here, these electrons which are passing through electron transport chain contain enormous amount of energy. And if this energy is released at a time, what happens? Much of the energy is lost in the form of heat and not made available to the living beings. So, the energy present in these electrons is released stepwise. Gradually it is released. And that energy is used in the ATP synthetase to convert ADP into ATP. That energy is also used to pump hydrogen ions from mitochondrial matrix into the intermembrane space. So the very purpose of electron transport chain is to release the energy present in the electrons in a slow and stepwise fashion and made it available to the synthesis of ATP from ADP. So in this way, the energy released here is used to convert ADP into ATP, and I already have told you for each pair of hydrogen ions, one ATP molecule is synthesized. So now take again NAD. NAD is entering the electron transport chain here. From here means 3 pairs of hydrogen ions are pumped into the intermembrane space from the matrix. That means 3 ATP molecules are formed for each NADH, reduced NAD. For each reduced FAD, only 2 pairs of hydrogen ions are pumped from here to here. So it causes production of only or synthesis of only two ATP molecules. That is the main difference between NAD and FAD. So in this way, these hydrogen ions which are entering through ATP synthase come here, electrons from there to come here, and the oxygen obtained through respiration is used here, and we get one molecule of water. In this way, water is formed and ATP is synthesized. This water is called metabolic water, and it is used by the plants and animals for their various needs, or it may be expelled. So this is about electron transport chain. One more important point about these complexes, major complexes, is in them reversible reduction occurs. These are all protein complexes. In these protein complexes, reversible reduction occurs. That means oxidation and reduction occur simultaneously. Then, Chemiosmotic hypothesis, I have explained. Electrochemical proton gradient is established between the intermembranous space and mitochondrial matrix. I have told you, because of the pumping of hydrogen ions from here to here, electrochemical proton gradient is established across this inner mitochondrial membrane. Because the oxygen is involved in this process is known as oxidative phosphorylation. Oxidative phosphorylation. Then, for each pair of hydrogen ions passing through ATP synthase, one ATP molecule is synthesized, as I have explained this. Then, coming to the respiratory balance sheet, how much energy is produced during respiration? During glycolysis, for each, here the result is for each glucose molecule. During glycolysis, two ATP molecules are produced and two NAD molecules are reduced. During the conversion of pyruvate to acetyl-CoA, two NAD molecules are reduced. During Krebs cycle, two ATP molecules are produced here, and 6 NAD molecules are reduced. 1, 2, 3. Then 2 FAD molecules are reduced here. I have told you, each NAD molecule produces 3 ATP molecules. Each FAD molecule produces 2 ATP molecules. So here we got 30 ATP molecules, here 4 ATP molecules, and here are 4 ATP molecules produced, 2 during Krebs cycle and 2 during glycolysis. So in total, how many we get? 38 we get. How many ATP is used in this process, entire process? Again, we must go back to glycolysis. In glycolysis, 2 ATP molecules are used for each glucose molecule. So if we deduct those two, the net gain is 36 ATP molecules. So if the glucose is completely oxidized during respiration, how many ATP molecules we get? We get 36. Our net gain is 36. If it undergoes only glycolysis or partial oxidation, how many we get? We get only net gain, only 2 ATP molecules. So here we should be very careful about gross gain and net gain. Gross means total number of ATP produced, that is 38. Net means if you deduct the ATP molecules used during the process, we get the net result. The net gain is only 36 ATP. That is about respiratory balance sheet. Then, the respiratory pathway is also known as amphibolic pathway. Generally, respiration is a catabolic process, energy releasing process. Anabolic process means building up, energy gaining. Here, energy releasing process. But the respiratory pathway is both anabolic and catabolic. So it is known as amphibolic pathway. That means respiration participates in both anabolism and catabolism. Then, which substances are used in the process of respiration? All proteins, carbohydrates and fats are used. But the preferable substances are carbohydrates, mostly glucose. We have seen glucose is the starting point of glycolysis. If glucose is not available, glucose is obtained from glycogen or from other carbohydrates. If all the available carbohydrates are used up, then what happens? The next choice of the body is fats. The fats are broken down into fatty acids and glycerol. The fatty acids enter at the level of citric acid level or at the CoA level, coenzyme A level or acetyl CoA level. So the fatty acids enter the respiratory pathway at acetyl CoA level, whereas the glycerol enters PGAL level of glycolysis. Coming to the proteins, proteins are rarely used in respiration, metabolic respiration, because proteins serve various functions in the body. So the body spares proteins only as a last option. And these proteins can enter as the Krebs cycle or the acetyl CoA level. And from acetyl CoA level, the fatty acids are also resynthesized in the reverse direction. That means the fatty acids enter the Krebs cycle at acetyl CoA level and release energy. Or whenever the fatty acids are required from acetyl CoA level, the fatty acids are resynthesized. In this way, the metabolic pathway is amphibolic pathway. Then, respiratory quotient. Respiratory quotient we get when we divide volume of CO2 evolved and by volume of oxygen consumed. So if you divide the volume of carbon dioxide consumed by, if you divide the volume of carbon dioxide evolved by the amount of the volume of oxygen consumed, we get the respiratory quotient value. This respiratory quotient value is the highest for carbohydrates. The RQ value of carbohydrates is 1. For fatty acids, it is less than 1. If you take for tripalmitic acid, it is only 0.7. The RQ value for proteins is 0.9. So, carbohydrates have highest respiratory quotient. That's why the preferred substances in respiration are carbohydrates. So that is about respiration in plants. Though we call it as respiration in plants, it is a respiration occurring in all the cellular and all the cells of eukaryotes. So it is also known as cellular respiration. Thank you.